Great. Thank you very much, Olga, for the kind introduction. I assume you can see my screen now?
Yes.
Excellent. Yeah, thank you very much for organizing this series. I think that's a great opportunity to make the best of the current situation that we're all in. And I've decided to make this talk and let me just, oh yeah, okay. Can you see my cursor as well, the pointer? Great. So I've decided to make this an overview talk because there are so many aspects of nanodiamond science and applications that I couldn't really decide which topic to focus on. And hopefully there's something interesting for everyone. Unfortunately, I won't have time to go into too much detail for each topic. But if you're interested, please feel free to ask questions afterwards. And also, please feel free to get in touch. As Olga already said, no matter where you are, we can always have a chat online. So not all nanodiamonds are created equal. I'll briefly explain in a second what I mean by this. But first, let me briefly introduce myself. So in the last 10 years or so, I've worked in quite a few different areas, as Olga mentioned. But today I'll very much focus on diamond and small diamond particles and the area of nanomaterials and how these nanomaterials can be used in biological applications mostly. But before I start, I would like to thank many or some of the people who made all the research that I'll show you now possible. I think we have a very great and kind international community of researchers, and here is just a small selection of researchers that I've had the privilege to work with over the last years. Some of them are online, including Vadim. Sherry, I think, is not online. Olga, Peter, who spoke, I think, at the last lecture. Jean-Charles Arnaud and his team in Paris. Alex Zaitsev, Alex Shames in Israel. Takeshi Oshima, who is a very close collaborator in Japan. Jan Jeske at the Fraunhofer, and recently also Adam at Hyperion Materials Technologies. But also Australia is a very great place to do diamond research. And here the top row are all people at my university, which is RMIT University in Melbourne. Brent Gibson, Andy Greentree, Kate Fox, Alistair Stacey, Dougal, and Stefania is also at RMIT. And more broadly, Melbourne is a great place. So we have quite a few other colleagues in Melbourne that do great diamond research. One of them is David Simpson. We work with closely, but also other people nationally, like Marcus Doherty, Neil Manson, one of the most senior researchers in Australia, and a great person to talk to, and also Lindsay, one of the biologists we work with. But this is where I'm located right now. So if you look at the planet from that point of view, it appears that there's not much else other than Australia, which is obviously not true. But in the current situation, that's not necessarily bad, given the current COVID pandemic. So I am located in the southernmost part of the continent, which in summer is great because it doesn't get too hot. And currently, it's also an attractive place to be because we've had 39 days without a single new COVID case in our state of Victoria, which is quite amazing. If you would have told me that a month ago, I would have said, or 2 months ago, no chance at all. And in Melbourne, so this is the Melbourne skyline. It's a pretty amazing city to be in and to live in. And our university is in the heart of the CBD. So this is where we do most of our experiments here. But the thing I actually enjoy most about Australia, living in Australia, is that not very far from the city, you have absolutely amazing nature, pristine places, and also amazing wildlife. So what you just see here, that's a 2-hour drive from where I am right now. So long story short, when we can travel again, you are most welcome to visit us if you're interested, for scientific reasons, but also for other reasons potentially. Okay, so let's jump into the topic that I'm actually meant to talk about. Eugene said it's going to be exactly 4 to 5 minutes. I'll try my best. I'll keep it fairly short, as much as I can. And but if I miss something, please feel free to get in touch after. So not all nanodiamonds are created equal. I think this is a statement that's true in a few different senses. So the simplest one is the literal sense, in a way that nanodiamonds can be made in different ways. And probably, or arguably, the most important ways to make nanodiamonds today is either from HPHT, synthetic diamond, or using a detonation synthesis. So this is probably something that many of you would know already. You can use high pressure, high temperature synthetic diamond, crush it, and create nanoparticles, which have fairly random shapes. And it's very difficult to make them really small, as in below 20 nanometers in size. Then we have detonation nanodiamonds, which are made from explosives in a detonation synthesis, and you get quite small particles that are only 5 nanometer in size or so, but have very different properties, irrespective even of the size. The properties are very different. So in that sense, nanodiamonds are definitely not created equal. And we're also interested in these particles for different reasons, generally speaking. So HPHT nanodiamonds, most people are interested because we can create so-called color centers, which are fluorescent defects in the diamond crystal lattice. The most famous one, obviously, the nitrogen vacancy center. And we are interested in these because they are single photon emitters. We can do quantum physics at room temperature. We can use them for sensing, magnetometry, thermometry as 2 examples, and they are extremely photostable. Now, detonation nanodiamonds, most people are interested in those because they are quite small. They have unique mechanical and chemical properties, and it turns out they're also fluorescent, but that's more of a niche application at the moment and not really well understood. So I'll talk a little bit about this later. And traditionally more used in completely different areas like drug delivery, composite materials, and also as abrasives. Nanodiamonds are also not created equal in a second sense that is quite fundamentally different. So even if you look at one type of nanodiamonds, in this case, HPHT nanodiamonds, these are a number of SEM images of these, of one batch of particles. So this is 500 nanometers. And it's quite obvious that each particle is quite unique. And this can be a challenge or an opportunity, and I'll explain later a little bit more what I mean by this. But in this sense, yeah, within each batch, not all nanodiamonds are the same at all, and in that sense, quite special and different from many other nanomaterials. Okay, so I'll start by talking about high pressure, high temperature nanodiamonds, and if time permits, I will also speak later a little bit about detonation nanodiamonds and applications. So why are we interested in fluorescent nanodiamonds? Well, one reason why many people, including myself, are interested in them is because they are fluorescent nanoscale sensors. So this is an image of a cell that you see in green, the cell nucleus in blue, and all the red dots that you see are fluorescent nanodiamonds. And what we would like to do is use each of these particles, for example, from magnetic field sensing, temperature sensing, or maybe even voltage sensing, or other sensing protocols that you can use here, or other properties that you can measure. So in this context, if the properties of each particle is different, that's an obvious challenge for application. So in an ideal world, you would like each sensor, each nanoscale sensor, to have very similar properties. For example, in terms of the sensing sensitivity, fluorescence, brightness, and so on. And I think that's a challenge for us, and something that we are definitely trying to change in various ways. But how do fluorescent nanodiamonds work? Well, pretty much like most other fluorescent nanomaterials, you have a particle, you shine light. In case of nitrogen vacancy center and nanodiamonds, usually green light, and the particles fluoresce in red. In case of HPHT nanodiamonds, usually because of color centers, like the NV centers inside the crystal lattice. And then the surface of these particles is relatively inert, which is great for biology, but at the same time, we can attach many different types of biomolecules to the surface, which is great for targeting specific things in biology. And potentially, most importantly, we can use these color centers for sensing, as I mentioned before. But first of all, the color centers need to be created, because they usually, well, they do not exist at high concentrations in natural nanodiamonds. And that's one of the main reasons why we've stuck with high pressure, high temperature diamonds, because they already contain nitrogen in very huge quantities. So this is a photographic image of a large, so millimeter scale HPHT sample that contains nitrogen, which gives it a yellow color. And when we create NV centers, this color changes because the absorption of the NV center is different. So the question is, how do we create NV centers in the first place? And as you probably know, there are many techniques of how to do this, and and so what you need to do is create vacancies, right? So we already have nitrogen. So in order to create NV, we need the vacancies. And a very common way of doing that is to use electron irradiation. So we start with a diamond sample that contains nitrogen. You irradiate it with electrons to create vacancies, and you perform annealing to enable vacancy diffusion, which creates the NV centers. And this is a project or a typical project where we work very closely with Takeshi Oshima in Japan, who has very amazing irradiation facilities, not only electrons, but also electrons. And this was led by one of our PhD students at the time, Marco. So basically what we found in this study here is that when you combine the irradiation and annealing step in one step, so you irradiate at high temperatures, you basically increase the generation yield of NV centers twofold. And happy to explain in more detail why we think that is, but don't really have time to go into this now. So this is nice. I think Peter already told you about a new irradiation technique he came up with a few years ago in his last lecture, which is where you basically put the particles in a boron 10 liquid into a nuclear reactor, and then use the alpha decay to create vacancies very efficiently. So the great thing here, to me at least, is that you get to a very high fluence within a few minutes, whereas in other cases it takes days, depending on the device that you use for irradiation to get to these fluences. And the particles still have quite good properties. But because there are so many different techniques that you can use to create vacancies, we wanted to look a little bit more closely at whether or not the irradiation type affects the NV properties in larger nanodiamond particles. So we know, for example, that the surface plays an important role. So we decided here to look at larger particles with a mostly oxidized surface containing many carboxylic acid groups. And we used many different types of particles that are available, some from Peter's lab, some from Olga and Adamas Nano. So these are from Adamas. This is FND Biotech. And basically try to investigate these samples systematically, in particular looking at individual particle properties as well. So what I did is I took an optical image of particles on a marked substrate and also acquired SEM images of the same particle to make sure that we're actually looking at a single particle, because otherwise, if you don't do SEM imaging, you don't know how many particles you're really looking at. And then we collected as much data as we could from this particle, including a fluorescence spectrum, lifetime, stability, and the T1 spin relaxation time. And yeah, that was published last year in this paper, if you're interested in details, but I'll just show you a few important things. So this is a typical fluorescence spectrum of an NV containing nanodiamond in a confocal fluorescence spectroscopy experiment, where you see the overall fluorescence of the particle in black, and that consists of two contributions from the two charge states of the nitrogen vacancy center, the NV- and the NV0. And the reason this is very, very important is because only the negative charge state can be used for the majority of sensing applications. And we used a mathematical model here, which is called non-negative matrix factorization, to really figure out what the relative contribution of those two charge states is for individual particles. And the interesting thing here is actually not what the average value is, but the spread between individual particles. So here is just this NV ratio. So basically, the higher the number, the more NV- you have, and the lower the number, the more NV0. And the different sample types that we looked at. And between the different irradiation techniques, we didn't find a systematic difference in this case. But for individual particles, you find huge differences. So some particles predominantly show NV- fluorescence for all samples, and some particles mostly NV0. And that's a challenge. And also some of the other properties, such as fluorescence brightness, fluorescence lifetime, the variations are quite huge between individual particles. And again, happy to discuss why that is. There are multiple possible reasons why this could be, but we're still trying to find a definitive answer, and more importantly, try to overcome this challenge. So the nitrogen vacancy center is not the only interesting color center, quite obviously. Quite recently, we have started to look at other color centers in collaboration with several colleagues internationally. So in particular, silicon and nickel-based color centers. Now, these particles were made at Hyperion Technologies, led by Adam Dallis in the US. But also Olga, Alex Shames, and Alex Zaitsev were involved in that project. And Alex Shames did very, very nice EPR analysis of those defects. And we mainly identified 3 defects in these particles. So in the silicon-doped particles, we found a silicon boron defect, where we don't exactly know what that defect looks like at the moment. Problem is that we can't detect it in EPR. So it's difficult to identify the structure. And the quite well-known silicon vacancy defect. And here most particles contain both centers. But what's nice about the material, about the silicon and the nickel-based material, they show very, very little NV fluorescence, irrespective of where you excite the particle. So you can excite them even with green light, and you will mostly see the silicon-related emission and the nickel-related emission. And for all of these, no irradiation is required. And in case of the nickel-based center, we were able to identify that center as substitution, the positively charged substitutional nickel defect, which is not trivial. So have a look at the paper if you're interested. There's quite a lot of discussion around which center is which in this area. But the nice thing from an application point of view is that these particles show very pure near-infrared emission above 800 nanometers, so between 800 and 1000 nanometers, which is a great spectral window for bioimaging. So we're continuing with this work and trying to make some use of it in bioimaging experiments. Another nice thing is that, so most of the work here was done on micron-sized particles, but the optical properties of these particles are still present in nanoparticles, around 50 nanometers in size. So for the silicon vacancy center, it was quite well known that they are happy that they survive in small nanoparticles, but for the nickel center, that was not so obvious. So this is fluorescent spectrum of the different particles in water. So you see the silicon doped particles here and the nickel doped particles here, the micron-sized particles and the nanoparticles as well. So for many applications, we need to put the nanodiamonds into something else. So it could be the cell directly if we're interested in bioimaging, and I'll show you a few images later, but often we want the particles to be inside a scaffold so we can use them for imaging or sensing. And this is a project that we've started last year together with friends in Naples in Italy, led by Luigi Ambrosio. And we integrated fluorescent nanodiamonds into polycaprolactone fibers using electrospinning. So here you can see an overlaid bright field image and fluorescence microscopy image of the nanodiamonds, which are in red here, and the fibers, where you see mostly larger fibers here in black. And if you look a little bit more closely, you can see that some of the fibers are very or relatively small, and that inside these, so this is a sort of a bright field image of the fiber. And if you look at the fluorescence from those fibers, the fluorescence background is extremely low of polycaprolactone, which is great. And this fluorescence here is exclusively from fluorescent nanodiamonds. And you can do optically detected magnetic resonance based sensing using these particles in PCL fibers, which is nice. Another material that my colleagues at RMIT have looked at, led by ASMA and also Brent Gibson, is a silk nanodiamond membrane, which we would like to use at wound dressings, which you could also use for temperature sensing. So it turns out that temperature is an important indicator for the health of your wound. So if it's not healing well, the temperature is raised quite significantly. So here it's just a proof of concept that this works. So you can make these membranes, and you can also do ODMR-based temperature sensing of membranes that have cells grown on top of them using the fluorescent nanodiamonds. We're getting to more difficult materials as far as fabrication is concerned, harder materials, and one of those that we're interested in is glass. So this is a project that is also supported by the Australian Defence Research Group, so DST, Defence Science and Technology Group, and led by Dongbi, Brand and Heike at University of Adelaide, where they have amazing fiber fabrication facilities. Now, the big challenge with getting diamond into glass is that glasses are made at very high temperatures, where even diamond particles will usually oxidize very, very quickly. So at 800 degrees and above. So here they came up with a little trick where we create the optical fibers that are doped with fluorescent nanodiamonds by sort of separating the outer and inner layer of the fiber initially, and put this into a tube and then draw the fiber. The advantage is that we only have to heat the glass for a very brief period of time, which the diamond particles survive. And the goal then is, and the reason why defense is interested, is because we would like to use these fibers for remote magnetic field sensing. Again, using, for example, ODMR. And another thing that we're trying to do, and this is a project led by Kate Fox, also at RMIT University, is try to use 3D printed fluorescent diamond particles as composites for biomedical implants. And here we used diamond with titanium, because titanium is currently used in many implants, but it has many challenges associated mainly with the interface between the biology and the titanium surface. And we know that diamond is a material that is usually very well tolerated by many types of cells. So here we've used a huge industrial 3D metal printer where we simply mixed the titanium precursor particles with diamond particles and found that the fluorescence actually, so the particles survive the process, and also some of the fluorescence is retained in these composites when you look at the surface. So that's quite interesting potentially from a surface material surface engineering point of view, but also potentially from a sensing point of view at this interface. Okay, so now another area is imaging of diamond particles inside cells. And one of the great things about fluorescent nanodiamonds is that their fluorescence is very stable, and you can see the particles in many different types of imaging modalities. And here in this paper, we basically did some correlative imaging, very difficult to pronounce this word, correlative imaging using fluorescence microscopy, scanning electron microscopy, atomic force microscopy, and also, as probably a slightly newer technique, soft X-ray tomography. And that was published quite recently, where we basically compare the different things that you can see or not see using the different imaging modalities. And it's quite interesting, and it shows you that in many cases just using one modality is just not enough to get a complete picture of the biological system that you're interested in. And the new aspect really here in this paper is that we were able to get very nice high resolution 3D images of nanodiamonds inside cells, so with sort of 20 nanometer resolution or so. So this is one of these soft X-ray tomography images, which we took at the Spanish synchrotron a few years ago, ALBA, with the great support of the staff there. And you can see the nuclear membrane cell nucleus here. And you can actually directly see all the organelles around the cell nucleus. And you can also see individual vesicles that contain fluorescent nanodiamonds in this case, which is really nice. And in the future, I think will help us better understand where exactly particles go inside cells in 3 dimensions. Okay. if I have a little bit of time, I'll also talk about detonation nanodiamonds. Yeah, how much time do you think? Yeah, okay, in time.
How much you use?
Sorry. Okay.
How much you use?
Okay. I'll keep it. I think I'll keep it short. So the physical and chemical properties of detonation nanodiamonds are quite different from high pressure, high temperature nanodiamonds, and I can't stress this enough. And I think as a community, we should mention very, very early on what type of material we're working on, because people outside our field may not know that these particles are very different, and we have to tell them. This is very, very important, I think. So, and one of the things I was actually very confused about when I got into the field is, why not use these small particles as fluorescent labels, right? They are so small and they're all sort of roundish shape. Great. Well, the problem is that the fluorescence from those particles is quite different. So are they fluorescent to start with? Well, I would say sometimes. And generally, the origin of that fluorescence is different from HPHT nanodiamonds. So many people have seen nitrogen vacancy center fluorescence from these particles, but at the moment, we have no reliable way of engineering color centers in detonation nanodiamonds. Please correct me if I'm wrong. I would be great if we could, but I'm not aware that we can. So you can always see color center fluorescence in a subset of particles, but the majority of fluorescence probably originates from the surface of the particle, as our group and many other groups have reported in the past. So one project we did a while ago with Vadim, who, as you know, is one of the leading experts in tuning the surface properties of detonation nanodiamonds, is that we just looked at what happens to the fluorescence as you change the surface chemistry of the particles. So when you buy detonation nanodiamonds from a commercial source and it's not particularly pure, you always see this sort of broad fluorescence from the unpurified material. When you really purify the material using, in particular using air oxidation, you can almost completely remove this fluorescence and the material becomes almost non-fluorescent. And then when you, for example, hydrogenate the particles, some fluorescence appears again, but the properties are actually a little bit different in the case of hydrogenated particles. And if you attach a molecule to the surface, which is absolutely non-fluorescent, such as this one here, I can't pronounce the word, but you can read it, the particles become hugely fluorescent. And Vadim has published a paper quite a few years ago now, which also shows this fluorescence, which is a lot brighter. And then you can look at a few other surface functionalities, and the fluorescence drops quite significantly again. So what we think happens in these particles, basically, unless you completely deaggregate them, you have the sp3 diamond primary particles that are always still a little bit clumped together into these aggregates. And you always have some fraction of sp2 carbon as well. And in these, we always see some NV fluorescence if you look carefully at enough particles, but most of the fluorescence comes from non-diamond carbon. So when you change the surface of those small aggregates, the fluorescence changes very dramatically. And the exact molecular origin or atomic origin of this fluorescence in many cases is also not fully understood, but many of the fluorescence properties are very similar to properties of material that people usually refer to as carbon dots. But carbon dots consist only of non-diamond carbon. So because the hydrogenated nanodiamonds had somewhat different properties, optical properties compared to the other particles, we tried to look at those a little bit more in a little bit more depth, and that was a project with Jean-Charles Arnaud, who is the expert on the plasma-based hydrogenation of the particles, and Vadim, who is the expert on the hydrogen annealing-based functionalization. And long story short, we did a lot of characterization on those particles in a lot of detail, including X-ray absorption spectroscopy, XPS, electron energy loss spectroscopy, TEM imaging, to find out where exactly the fluorescence comes from. But long story short, the fluorescence is very similar, irrespective of what you do or which functionalization technique you use. And we still could not prove definitely that the fluorescence for those particles comes only from the surface. So it's still possible that some of the fluorescence we see from those particles actually comes from NV centers, which we know are present inside the particles, but just may not be fluorescent. Another question is what happens when you completely deaggregate these small aggregates to the primary particles, which where Vadim and others have developed quite a few cool techniques to do that. And again, long story short, these particles in many cases are still fluorescent. And a while ago, also with Olga and her team, we published a paper. So if you buy the particles from Olga, the fully deaggregated particles, they will be quite fluorescent. And here I looked at the pH dependence of this fluorescence. So it turns out pH changes or quenches the fluorescence as the pH decreases of those particles, again, showing that this fluorescence definitely comes from the surface of the particles. And you can use these particles for imaging. The fluorescence is not as photostable, which is potentially a drawback, but you can get high resolution images like these. In this case, 5 nanometer particles used in breast cancer cells, also led by Olga and her team. What exactly gives rise to the fluorescence in these fully deaggregated particles? We're still trying to figure out. Again, an effort involving Sherry Chang, who is now back in Australia, Vadim and Olga, where we look at different types of these deaggregated particles, where here we have 2 types, and you can look at the TEM images that are fluorescent quite strongly, and other types that almost show no fluorescence at all. And the interesting thing is, we did most of the characterization techniques we could think of, including, again, X-ray spectroscopy, NEXAFs, Raman spectroscopy, FTIR spectroscopy, ICP mass spectroscopy. And looking at all of these, we can't really find the main thing that makes the difference or that causes these particles to be quite fluorescent and these particles to be non-fluorescent. So the devil must be in the details here, and we really we must understand really the atomic structure, in particular of the non-diamond carbon that is still present at the surface of these particles. And that's something that Olga has also reported in her papers a long time ago. But also something that's really interests me a lot is the colloidal properties of detonation nanodiamonds, but increasingly also fluorescent nanodiamonds. And this is a project that was led by Sherry, and we published a paper earlier this year in Nanoscale, where we looked at the self-assembly, dynamic self-assembly of these deaggregated detonation nanodiamonds in water. And Sherry took some really cool cryo-TEM images that show these amazing structures that you can see here in vitrified water. And we know now that this is definitely a dynamic process. These structures here form dynamically. So when you disrupt them, they will form again in different ways, and they probably evolve over time as well. So this is quite interesting. And there are a few hypotheses why that happens. But have a look at the paper. But what we are doing at the moment is looking a little bit more detail at the concentration dependence and the salt dependence of this dynamic aggregation, in particular using dynamic light scattering and small angle X-ray scattering, or SAX, which is a quite powerful technique to get a little bit more insight into the structure of the particles really in water. So rather than looking at a vitrified sample, we can look at water itself, which is great. And what basically, long story short, this is unpublished data, what we find is that at low concentrations, the particles sort of arrange in a more disc-like structure and a very fluffy fractal. And when you go to higher concentrations, they tend to form more elongated rod-like structures, and they become more condensed. And this is an effect that I think many colleagues have seen when they do DLS, where apparently when you look at the dynamic light scattering of highly concentrated detonation nanodiamonds, they appear to be smaller in your DLS experiment. And we think that this dynamic aggregation and the concentration dependence of this aggregation is the main reason that we've all seen this. So hopefully we will be able to publish those results fairly soon. And that was led by a PhD student in our team, and also by an expert at RMIT on dynamic light scattering and X-ray scattering, Gary Bryant, and experiments were done at the Australian synchrotron. All right, I've covered probably too many topics already. So with this, I would just like to thank the team at RMIT. That's a bit older image because we haven't been able to get together very recently, as most of you probably, with your teams. And would like to thank the various funding agencies for their support, and also companies for their support. And last but not least, thank you very much for sticking around at the sometimes not ideal time where you are located right now. So thank you very much.